I welcome you all once again. And today's lecture session, the speaker is Professor Biotechnological. I think we don't need to introduce him because I guess everybody knows him very well in the current office. Professor Sharma is the director of our DBT-AAU Center. He did his master's from our university and PhD from IIT. His research is mainly, he started with tissue culture of medicinal plants, but then he switched over to genetic transformation of brain vacuums. And he has two major achievements in the research area. First one is he developed transgenic that is resistant to stored red cells called proteins. The second achievement was his the BTGP, so he has developed two different BTGP lines, one having the primary gene, the other one with the Category gene. And he, after developing the transgenic lines, he didn't just keep it in the laboratory, rather he actually tried to take his material to the field, either directly or through some partners. So he established a public-private partnership with the Maharashtra Hybrid Seed Company. The Hybrid Seed Company is one of the major companies in India who sells hybrid seed in India, including the BT cotton. They are actually involved with the BT cotton release in India. And he also able to establish collaboration with the Punjab Agricultural University, University of Agricultural Sciences, Harvard, the IIT, IIT Kanpur. All these institutes are now collaborating with us because of this BTCP lab. After this achievement that he had with the BTCP, the government of India, especially the Department of Biotechnology, actually decided to fund us generously to have a separate DBT center, and with that fund we established this center. So he, under his leadership, we were able to manage to establish this center. And then I'm happy to say that because of this very good leadership, we were able to get more additional funding. Initially, the center was actually a DBT-AAU center, called as DBT-AAU center. Then we received additional fund for the DBT, and now the center is called as the Northeast Center for Agricultural Biotechnology after receiving those additional funds. So he has done a lot for our department, so we are very much indebted to him, and his contribution is also a very good speaker as well. Today you will be able to know how nicely he presents his research work. So I welcome Professor Sharma to the audience. I think he is very good.
Can I have that microphone?
I'm sorry, I forgot to mention one of his achievements. May I, sir? He's a recipient of prestigious Nobel Gold Chair in International Agriculture with a national professor position by ICR in March 21, and he was a national professor in 2021.
First slide, this is the second slide. No, this is the number 2. First one is different. Who is going to? It's not working. What? Hello? Is it working? I don't think so. It's without microphone. It's with microphone. Is there any difference? Little bit of it. Okay, that's fine. So, pointer is here. I didn't want Sumita to be here, but she is here because she has been hearing me for the last 35.
Actually, he's telling I'm old enough, and I pretend to be deaf, and I'm going outside. So.
No, it's okay. He's busy. I'm relieved now. Okay, how many of you have listened to me before? You have heard me before? 1, 2, 3, 4, 5, 6, many. So for you, some of the slides will be definitely repeated slides, I think. So please excuse me for that. I have to show those slides as well because many of you have not seen them. Okay? So I kept the topic as development of climate smart crops to the biotechnological intervention. Yesterday, in my speech, I mentioned that there are many constraints, okay, in agriculture which cannot be achieved through conventional technology, and that is why we need the biotechnology interventions. Before that, I'd like to know how many of you are from biotechnology background? Most of you are from biotechnology background, right? And how many of you are from botany zoology? All right. So anyway, so you are good at genetics, right? Genetics and biotechnology both, I guess. All right. Okay, next slide, please. When we, oh sorry, I have to tell myself, right? Okay, it's okay. two questions, important questions in front of us. We can answer. What are those? How can you produce more and more foods for the expected 2 billion population by 2050? That's a big stage. Okay. And next question, because of climate change, which is enhancing various stresses, whether it's abiotic stress, whether biotic stress, stresses are getting enhanced because of climate change. So that's why that is giving more threats to the agriculture in general. Okay. So these two are big questions in front of us. So what I'm going to discuss today, based on that, whether the biotech intervention is necessary for the development of climate smart crops. First question I'm going to address. The next question is, can genome engineering, we have a man from genome engineering, Sumita and myself, that she will be speaking to you this afternoon. So, genome engineering, the modification of genome, whatever is the technology, whether it's transgenic technology, whether cisgenic technology, whether genome editing technology, but as a whole, it's the genome engineering technology. So whether the genome engineering, because I cannot discuss biotechnology in general in 1 hour. Okay? So that's why I have chosen my own field. Can genome engineering technology help develop climate smart GM crops? And this I'm going to discuss with my own research. Sumita has already mentioned that. And soon we say yes to the GM crops. Here, the leaflets for you. And you can have one copy. Wherever I do go, I just distribute these things, this particular leaflet, because we are tired. We are tired. Why we are tired? Because we have developed our important chickpea lines resistant to insect pest about 10 years before the lines were ready, but still we are starting to bring them. Doesn't matter. They can, you don't have to distribute. One by one they'll take. I have many more copies. If somebody is not getting, that's fine. Okay. So third question is, should we say yes to the GM crops? So obviously I'm going to discuss what is the global acceptance for the GM crops. How many countries are accepting it? And if there is any threat to the human health, all these things we will discuss today. Okay. Well, the first slide shows the first statement I have put there, why biotech intervention to develop climate smart crops. As I said about the population, you all will agree, right? We have to feed more and more people. Food grain production needs to rise by 70% to meet demand. One picture is enough, I think. Oh, you are recording the whole thing? Okay. I have to be careful then. Now you see, elevated carbon dioxide level, see 550 ppm by 2050. That's the statistics shows. It's likely to reduce the nutritional quality of crops as well. So we are at the tight spot. You see, not only increasing the production, but we have to increase the production of the quality food. Earlier people are not very much concerned about the health, but today we are all concerned about the health. We are all concerned about the environment. So enhancing the crop yield and then quality food, big challenges. And uncertain floods, droughts, heat and cold challenge the crop yields. So conventional agricultural practices cannot meet such demand. Why? Because to undertake conventional breeding, we need the genotypes having such traits. So within the gene pool, in many cases, within that same gene pool, there are no such genes available to undertake conventional breeding program. That is why we have to go for biotechnological intervention. Well, biotech crops are adapted to climate change. Why have I said like that? Because these crops can be modified faster through biotechnology than conventional breeding approach. Because in a conventional breeding approach, you need long years to get the genes at homozygous conditions. But here, the regulatory process is a different thing. See, we have chickpeas resistant to insect pests in hands. We have, but we are facing some regulatory hurdles. That's a different. Sure. But for the development, obviously, it takes much shorter period than the conventional breeding program. And why we need to develop them faster? Because of the climate change, insect pests as well as diseases, these are occurring continuously with their variations. Newer and newer insect pests are coming. Newer and newer diseases are coming to us. That's why we need to develop them, the alternate forms of the particular crop have to be developed, within a short period of time. That's why also the biotech interventions. Then resistant varieties will reduce the pesticide application and hence reduce carbon dioxide emission. You see, once you have the insect resistant lines, obviously you will spray less amount of pesticides. That will save your diesel consumptions. That will save the carbon dioxide emission. You get my point? So this will definitely these are good for the environment as well. They will be environment friendly. And crops tolerant to various abiotic stresses have been developed in response to climate change that I'm going to show. This is, you see, one of the examples that herbicide tolerant, GM crops. Soybean is there, canola is there, cotton is there. These are commercially available lines, resistant to herbicide. How this will help? How this will help the agriculture, you see? First thing, because once you have the herbicide resistant lines, a variety, for example, then for the why do you spray the herbicide? To kill the weeds, right? Weeds are havoc in many crops. So if you can spray the herbicide, then you have to make less tillage operations. Tillage operations will be lesser because we do the tillage operations to reduce the weed infestations. Have you seen how many of you are from the farming family? How many of you are? 1, 2, 3, at least few I have seen. Okay. Have you seen, a pulse cultivation? Say, background? Huh? No? You have seen. So, I have a very bitter experience, with why is, before discussing this slide, should I tell you a story? Okay. Okay, story is there. In fact, the farmers have to struggle a lot, whether it is tea cultivation, whether it is chickpea cultivation, mainly the upland crops, which infestations is very, very much. When we were in the third year of BSc Agriculture, there's a course, it's called Practical Crop Production, PCP, Practical Crop. Is there anybody from Assam Agriculture University here? Okay, PCP, Practical Crop Production. Here we were given a land, okay, big land, and four students, and then we were given the background seeds. We have to cultivate it, sow it, plant it, and then we have to up to the harvesting, and we have to show, what is the product, how many kgs we have produced from the, crop. That's all. That is the practical crop production. So in my group, I was from Guwahati. I have never seen in the rice crop. But while coming from the past, sometimes we used to see, but not very closely. I have never seen maize. I have never seen sugarcane. I have never seen black gum. It's like that. We are not from, unfortunately, we are not from village. So, other three guys also of the same categories. One from Jorha, and one from, I think, Balagna or something, and one from Nagaland. Okay, so what happened, seeds were given, then anything can be managed. This is a big, tactic. You can manage. So somehow we managed to prepare our land. Land got prepared, very nice. We managed. Then, seeds were sown. We are very happy. But, it's so nice. We sowed the seed. We went back. After a week, we just came to see the crop. We went a little closer and we saw very green crop. It's coming. We're happy. We didn't go to the closer. We went back, had char, samosa. Then after another week, we went. After a long distance, we saw, it's nicely good. Further, we went back. Again, chai, samosa, and rasgulla. Then, after another week, we went, and from very long distance, we saw all other plants didn't have any greenness. But our plants had heavy, was green and green. Lots of things were green. I asked my friends, have you done something? He said, no, I have not done anything. Nobody did anything, but why it is so big? We were enthusiastic to see those. We went closer. Okay? And then we saw only one black drum plant there. All weeds. So that's the scenario of weed infestation. That's why if you have the GM crops like soybean is made herbicide tolerant, and you can spray, farmers can spray, weeds. I say herbicide. Okay? And you can see here with only soybean in the US, if you see, permanent fuel saving, fuel saving is 835 million liters. And then potential additional carbon dioxide, saving is 2,295 million kgs. That much of carbon dioxide you can save on the herbicide. So this is how it is helping us. This is the we call it as conservation agriculture. So see, GM crops are helping in organic agriculture. How? The less spray of pesticides. We are supporting, in fact, we are helping the organic agriculture. In the herbicide tolerant crops, less tillage operation, less amount of soils are getting lost. So that's the conservation agriculture. Okay? So, see, these are some of the transgenics for abiotic stress tolerant transgenic lines. There are many, many examples, but I'm just bringing a few of them. So rice for drought, salinity, and cold. Tomato for temperature and drought tolerance. And these are mostly, the genes are most the transcription factors. The transcription factor is introduced into the crop through genetic engineering. Okay? Similarly, in cotton also, we have now heat tolerant cotton. This has been recently developed. And you see, genome editing, you might have been hearing genome editing for quite, number of times might be, right? Because this is the most advanced technology today to modify the genome. So, but the question is that in our region here, we have just started genome editing project. As I yesterday mentioned, 3 projects. One is Sumita, one is Sarvinder, and one is Ratna. There are 3 projects going on. At least we should be proud of them because this is the most advanced technology for modification of genome, and Assam Agricultural University is having already 3 such programs. But see, the US already had genome edited plants in the field in 2019. So that is the soybean. First one, you can see soybean with low unsaturated fatty acid. It's a big problem in most of the oilseed crops, unsaturated fatty acids. But they have produced a lower unsaturated fatty acid soybean. So that has been made through genome editing, and by editing these 2 genes, and then these lines are already in the field in 2019. I'm not going to show the other example. There are many examples, just we can get from the internet. Now, great news is that here, not like you should be very happy now. Okay? To me, I was very, very excited because we are going to produce the genome edited SGN1 and SGN2 kinds of plants. SGN means 5 directed nucleases. 1 means just you can change, substitute 1 base pair. That can lead to a new phenotype. That's SGN1. So you don't have to add any foreign DNA there. This is just like a conventional mutations. Only thing is that genetic engineering technology is used. It's a CRISPR-Cas technology or talent technology or gene printer technology. These are the different ways to modify a genome, very a genome. So here in SGN1 or SGN2 means a few bases you can add, of course, but not a whole gene. In transgenic or cisgenic, we add a complete gene. Okay? But, so good thing is that this office memorandum in April 30 from the government of India, Ministry of Environment, Forest and Climate Change, then they have exempted the genome edited plants falling under the categories of SDN1 and SDN2 from biosafety assessment. There have no regulations. They'll be released in the field without having any regulations. So it's big news for all the genetic engineers. But the question is, there are certain traits that the crops need. For example, insect resistance in coffee, insect resistance in chickpea that I'm going to discuss. Genome editing you can do only when the similar gene is available within the genome. And only you can just edit it. If you are susceptible, you can edit certain base pairs to make it resistant, for example. So the gene has to be there in the gene pool. Then only you can go for genome editing. But if the gene is not there, for example, in the case of coffee, bond resistant coffee, the gene will have to get from a bacteria, right? Bacillus thuringiensis. But then, in that case, the transgenic is the only way. So you cannot solve each and every problems with the genome editing. So I'm sure as the genome editing has been allowed now, eventually, maybe in the next 20 years or 10 years, maybe the transgenics will also be in the field in India. And then there are several transgenics in field already in other countries also. Okay, so my next part is that genome engineering research at Assam Agricultural University for the development of insect resistant chickpea. Why? Why should we go for GM chickpeas? That's the first question. Okay? Why do you need to go for? Why we cannot go for conventional planting? As I say, genes are not available in the gene. You can see the 2 big problems. One in the field. One is in the field. This is the lepidopteran paste. See? And it goes inside the pod and eat every beautiful stuff inside the pod. So you can have 100% crop loss in this case for your problem. This is in the field. And this is another problem during storage. I think you might have seen, all of you might have seen, right? Such things at home. Is there any pure vegetarian here? Pure vegetarian? You are a pure vegetarian. Oh, I didn't know. You are a pure vegetarian. Okay, I didn't know. Okay, you are in the Northeast for how many years? Maybe now 8 years, 10 years, right? You're a very strong person. In the Northeast, it's very difficult to remain as pure vegetarian. Anyway, but don't think that you are pure vegetarian. Why I'm telling you now? You see, how these holes are developed. This is a coleopterian paste, coleopterian bouquet, okay? And this bouquet lays eggs. You know, these are white stuffs of the eggs. And after hatching of the eggs, the tiny larvae, you never see, you know, with your naked eyes. These tiny larvae, they go inside the seed. And the whole metamorphosis that is from the larval stage to the adult stage. This is the adult. Larval stage to the adult stage actually takes place inside the seed. And when the adults emerge, then they make the hole. See? So, your mother or you throw away these guys. Nothing is there inside. When there are holes, you'll be just throwing them out, right? While cooking are the beautiful chickpeas, and the beautiful tomatoes will be inside all these larvae. Okay? So don't claim that you are pure vegetarian. Sorry about that. But anyway, so what happens, you know, because of these problems, these are two major problems, and because of this storage problem, what, you know, the sellers they do, they apply herbicide, actually pesticides, no. They radiate the seeds, they apply pesticides, and in the large data in Assam, why I chose this problem? Because in Assam, there are, you know, lot of news articles coming. News, okay? That, you know, many people, they got affected because of, you know, eating chickpea in large gathering. We worship chickpea, I mean, God, you know, with chickpeas, right? All numbers, mandis. And in the large gathering, if you do not wash them properly, then there will be a lot of pesticides actually remain. And that makes, you know, that's the dead adult. Okay, so what I thought, these are two big problems, you know, long back. When I started, you know, doing my research after PhD, I thought, you know, I was asked to do a research in legumes by an American, you know, company that gave me, you know, a fellowship to go to America. And they said, after you come back, you have to work in chickpea. That was the condition. Then I thought, there's a story actually. Then I thought, you know, if I have to work in chickpea, you know, I must choose a problem. Genuine problem, you know. And I have to do that genetic engineering as well because they will train me in genetic engineering. So I have to do the chickpea research, this genetic engineering for chickpea to make something, you know, good for chickpea. Right? So I discussed with the farmers, I discussed with the breeders, and I came to know that these are the two very serious problems, and these problems cannot be solved in a conventional way because there are no insect-resistant varieties. So I took the genetic engineering. And then for genetic engineering, you know, first thing is that you have to have a tissue culture institution. I myself did my master's in tissue culture, PhD also in tissue culture related things. You know, biotechnological, of course, but you know, tissue culture was one of the aspects. So this is because, you know, we can introduce the gene in a tissue only, right? From that tissue, we have to regenerate the whole plant. Next trend we call it as. So for chickpea also, we developed a transformation protocol, in vitro regeneration and transformation system. You can see here, the experts, and then, you know, we just select them, you know, in the regeneration and selection medium, and then experts having the good shoots. Now we just select them, and then, you know, we root them. why this guy is, shouting. You see, sorry, what happens? Sorry. This is an in vitro grafting that we follow now. Krishna is a good transformation expert here. So she does now. This is grafting we do because there is a transgenic scion and this is the non-transgenic rootstock. Because rooting directly in tissue culture was difficult for GT. That's why I developed this in vitro grafting system. And then we established them in the soil. Okay, this is in brief. And then with this development, the transformation protocol once we have in our hand, then we have to think about how to make chickpeas resistant to storage space. Being from Assam, I took this as my first problem, storage space problem. Okay, and what have you seen? Are these chickpeas? No, it's rajma. You like rajma? You like rajma chop? Huh? Aapko bol pate ho na? Rajma chop. I don't like, but it's unfortunate. Whenever you go to Delhi, I समझ गया और यहाँ खाना खिलाएंगे रसमा चावल But I like rasma because they are resistant to storage space. I love it because at that time, when I started my research, then I came to know literature search is so important. I searched literature. Where did I get gene? Then I found one Australian group and one American group. They collaborated and they found the rajma, the kidney bean, or the stomach bean, or kidney bean, the red ones, okay? Deep red ones. They are having a particular gene called alpha amylase inhibitor gene. Amylases. We need amylases for what? For digesting starch. Insects also need amylases to digest starch, right? So amylase inhibitor protein, if you have in the food, then that amylase inhibitor will nullify the action of amylases, and insects will not be able to digest, starch, and they will die. So that's the story. So from rajma, we cloned the alpha amylase inhibitor gene from rajma, and we have to introduce that gene into chicken. Okay. Now you see, this is the genetically engineered, a tDNA. tDNA you know, right? Left border and right border, and here are the various, segments. And here is the alpha amylase inhibitor gene, and we drove this gene with a promoter called phytohemagglutinin promoter. Why we took this promoter? Because this promoter can express the gene in seed only, mature seed, because phytohemagglutinin is a stored protein, storage protein. We took the promoter from that because we want to express the gene only in mature seed, not in other plants. Okay, that is why we took this, seed-specific promoter. We call it as a seed-specific promoter. And NPT2 was used as a selection marker gene. Okay, and you can see. Once we introduce, following our transformation protocol, once we introduce it, and then we got southern blot analysis. We had to do a southern blot analysis to show whether gene got integrated or not. How many of you have done southern blot yourself? Is there anyone who has done southern blot by themselves? Are there anyone? No? Hare Krishna? You have not done that? I never say, how many Krishna students. We are saying no. And then you don't have southern blot? Okay, so see, our lab, we do the southern blot, but of course you do not. Why I have asked you? Because the southern blot is invented by E.M. Southern. E.M. Southern, perhaps you know, right? So E.M. Southern visited our lab in 2008. He and his wife visited our lab, and he was so impressed that not this lab, then we were in a small lab. From a very small lab, we today. And Southern and his wife, they have a trust, clinical trust. So they funded us, seeing our, developments, they funded us for background transgenic development. I mean, Sumita was a PI. So we got this, because trust funding for 5 long years from Southern. Okay, the same Southern plot analysis, huh? So here you can see, this is the control means untransformed chickpeas. And in Southern analysis, if you see one band, means there is integration of only single copy of the transgene. And 2 bands means integration of the 2 copies of the transgene. So what you have seen here, like these are untransformed chickpeas, and these are transgenic lines. And some of the lines have single copy of the transgene, and some of the lines have 2 copies of the transgene. And same lines, when you did the Western blot analysis, Western blot is for the proteins, whether they have the alpha amines in the protein or not. Here is my control. You can see here, no bands obviously. And in my transgenic lines, wherever there is integration of the gene, because it is important, gene we introduce has to be integrated in the genome, first thing. And second thing, that has to get expressed. Then only we will get the protein in question, right? So, all genes, in all lines, the protein was expressed. And you see, these 2 lines, 7 and 13, these 2 lines, we didn't see any band. That means they were just escaping the selection process. Somehow it happens in this process. Some of the lines, they get escaped. Although you put the selection, but they will escape. And this is the proof here. You see, they didn't have protein also, and they didn't have any. Very clean, Southern blot and very clean Western blot. But how many times we repeated, this I think we repeated for 20 or 25 times, then only we got such a cosmetic, stuff. Anyway, after doing the Southern blot and Western blot analysis, our next is, see, when there is a single copy, why I asked the genetics, you know genetics, right? If the single copy of transgene, what's the segregation ratio? The next generation. What's the segregation ratio if there is a single copy of transgene? Yeah? It should be Mendelian segregation 3:1. And if there are 2 copies, then it should be Mendelian ratio of 15:1. So you see, how do we confirm? These 2 lines, number 16 and 17, if you remember last slide, they were having the single copy of transgene, and we looked for the segregation in the T1 generation, and they segregated at a 3:1 ratio. So, unless we show that, single copy, if we have found this, or I have shown only 10 lines or 8 lines, but Sumita and I developed maybe more than 200 lines. Out of that, these were the good lines for us, because they showed that this is called a stable transformation. Stable transformation is one where we have the transgene integrated in the genome, where it is showing the 3:1 ratio, and also it is showing the exact phenotype that we expect. That is a stable transgenic line. So to get a stable transgenic line is very difficult. But anyway, this is the final slide for this story. See, here are the control rules, means without transformation. Seeds without transformation, chickpea seeds, and these are the 2 of the transgenic lines, 16 and 17 that I showed. Actually I named them later on. And you can see they are completely protected against the insect pest. Complete protection against insect pest. This is the storage pest. Okay? So this is the first success story from this laboratory, which we did in collaboration with CSIR of Plant Industry Australia. Okay? So after this success story, when we published this success story, then Government of India, as well as some foreign funding agencies, they came to us. They say, storage space is a problem, no doubt, but this problem is mainly in the eastern part. Okay? Because this is the part the chickpea is eaten after cooking on. So you should develop what borer resistance, which is the major havoc in chickpea cultivations, what borers. Okay? You should develop the what borer resistant lines. So for that, we thought of using the BT gene. Because at that time, already the BT cotton was in the field, 2004 it was. It was 2004. BT cotton was already in the field. BT maize was already in the field, so we know we knew that pyrazine is effective to control the left phase. Okay? So we use the same pyrazine, Cry1AC and Cry2AA. These two are the pyrazines we use. And you can see here, I'm not going to discuss this in detail because time will not permit me. But you can see there are two tDNAs. We call it as a twin tDNA binary vector. Twin tDNA binary vector. Twin means there are two tDNAs. In one we put the selection marker gene, and in one we put the Cry2AA gene. Okay, or PGC. So that we can segregate out the selection marker from the gene of our interest, that is BT. Because at that time, the human crop for using the antibiotic resistant genes. You cannot use the antibiotic, you cannot keep the antibiotic resistant genes in the crop. There was human crop, perhaps many of you have heard about it. But today it's not there. So that's why we develop our construct like that, a twin tDNA binary vector. This is the co-transformation. And what the promoter we use, that is the small subunit promoter of from Arabidopsis. Okay, small subunit of rubicicol from Arabidopsis that we do the promoter. This is a green tissue specific promoter. Green tissue because pod borers they infest all the green parts of the plant, not the mature seed. That is why instead of a constitutive promoter, we use a green tissue specific promoter. And then here is the PCR. You can see that these two are the controls, and with the simple PCR first we saw if there is presence of the gene. Yes, in the transgenic line there was. And you can see this is the Western blot analysis again. Here is the BT cotton we took as a positive control, BT cotton as a positive control. Because that time BT cotton was available with us. So we took that as a positive control, and this is negative control, means the untransformed chickpeas, and these are our transgenic lines. And you can see that line number 6H, 6L, these two lines, and those 5G, they were very high expression. There was high expression of BD. And in certain lines, like 5A, there was a little bit of medium kind of expressions, and in this one, very low expression. Why do you get this, variation in expression? Because same promoter we have used. Obviously, the integration of gene was in a different position. Depending on the position effect, there can be variation in the expression. And that is why we will get different kinds of lines. 이번 뭐 클래스라고 해서 비숍 공부했으니 어렵게 공부했어. And you see, as I say, that the pod borer infests various parts of the plants, right? For example, you can see the young leaves, old leaves, sepals, petals, pod walls, immature seeds, right? All these things are getting infested. So that's why we wanted to see whether the BT expression was there in all these tissues. You can see here, young leaves, there was expression, old leaves, expression, sepals, petals, and pod walls. Immature seed also, a little bit of expression was there. And in mature seed, almost no expression. Because the promoter we use was a green tissue specific promoter. Okay? And here is the southern blot. So 6H, 6L, if you remember, they had a single copy, they were high expression of BG, these two lines, and we chose these two lines because why? There was again single copy of the transgene integration. Okay? And same 6H and 6L, what I'm showing, because they are segregating at a 3:1 ratio. So again, for BT also, we got several transgenic lines. Single copy transgene, segregated at a 3:1 ratio, good level of expressions, right? And let us see whether they are insect resistant. Yes, you can see these are the leaves from we did the leaf bioassays. This will just pick up the leaves, okay, from the plant, and then we can do the bioassay, insect bioassay in the laboratory itself. So lab bioassay. The lab bioassay, what we saw, like this is a parent, and this null means negative segregants. Negative segregants we call it as the null lines. So non-lines and parents, they were, that when the insects were fed with those leaves, insects were very happy and leaves were very unhappy. And then here you see, this is one of our transgenic lines, high expressing transgenic lines, leaves were so smiling. You see, they are smiling. They are so green, healthy, right? So we are very happy at that time. Not only we, when we publish this, transformation system for the BT in chickpea, not only we, the funding agency was very happy. And funding agency wanted to transfer this technology to other institutions at that time. So they requested us to, showcase our technology. You see, many a times, we scientists, we develop certain things, but we are stuck there. We do not know what to do with this. So commercialization of your technology is also important as well. So what we do, we showcase our technology, and Maharashtra Hybrid Seed Company, as Sumita has mentioned, is the biggest, one of the biggest seed companies in the country. So at the same day, there was ceremonial transfer. You see, because this is the breakthrough. This was a breakthrough from Assam Agricultural University in collaboration with CSIR University of Australia. And of course, NIPB being Newland. Numbers of institutes collaborated with us. You see, we should have a problem, a serious problem. To solve the serious problem, whatever technology is required, what kind of labs, infrastructure is required, everyone will help you. This is the case you can see. We started from a very small lab, but at this point, we were with the BT chickpeas resistant to complete protection against the phosphorus, which was a breakthrough. And because of that, Government of India wanted a ceremonial transfer of technology. Vice Chancellor, our earlier Vice Chancellor, he transferred the technology, and the Minister of Science and Technology, and then Secretary, Department of Biotechnology, Government of India. And we transferred the technology to Maharashtra Hybrid Seed Company. This is big Barwale, 2 times winner of the World Food Prize. You see, Mahiko, the big Barwale, he won World Food Prize 2 times. Okay, so such a company took our technology, and later on we transferred this technology to Sangro Seeds, New Delhi as well. And you see, on the same day, because of politics, same day, not only to Mahiko, we also transferred to the public institutions. It's politically you have to see all these things. That not only to the private company, because while transferring to the private company, university earn money. I earn money. Okay? Because they paid us for having our technology. But at the same time, we transferred to US Harvard, University of Harvard. Because why we transferred? They had a track record. University of Science, Harvard had a track record of working in BT chickpea. BT brinjal at that time. BT brinjal was also going, BT papal was also going on in India. Okay, so they had a track record of working in these crops, in the BTG and in BTG. That is why we transferred our technology to US Harvard as well in the same day. And Mahiko moved very fast. They did the field test in 2014 and 15. 2 years of field test. I visited this field test myself. And you can see, with this field test, what they confirmed that there was a 25% yield advantage obtained. 25% yield advantage in the transgenic lines compared to, non-transgenic lines. Mahiko basically, in progress is seen into other varieties because we developed the transgenic lines in one particular variety. Okay? And Maharashtra Hybrid Seed Company transferred these lines to other important developers of that region. And this field test was very promising at the time. And after that, we thought of transferring to other public institutions. Now we are planning to transfer to many other public institutions. So in 2010 we transferred to ICRISAT. In 2011 we transferred to IIT here. In 2013 we transferred to Punjab Agricultural University. And now Punjab Agricultural University and US Harvard, they are moving quite ahead. Using our transgenic lines, they develop all the introgressed lines. As I said, you have to introgress this gene by conventional breeding approach. You see, once you have the transgenic line in hand, then you can follow simple breeding procedure to transfer this gene to other important chickpea cultivars. Okay, so you can see here, these introgress lines are doing pretty good, and we have the information now that the introgress lines were completely protected against the potholders. That's big news. And government of India and ICR, Indian Conservation is emphasizing us that this publicly developed transgenic lines should also be brought into the field. What we are planning now, Sumita is writing that proposal, and hopefully in July we will submit that proposal, right, for field days. So these lines will be now sent for the field days. Okay? And one very important thing is to satisfy the regulator. As I say, hurdles are there. Although we have the lines, but the regulators raise many questions. One of the biggest questions is that whether your transgenic lines are clean. Clean in the sense where it has gone to, we have to confirm it that this particular thing has been integrated. So southern court can say it is integrated. But where it is integrated, that is also a big question. Which part of the genome it has been integrated? Which chromosome it has been integrated? That you have to show. You also have to show that because of this integration, there was no alteration in other traits, especially the flanking regions. Genes in the flanking region should not get modified from its trait. Okay? So if Sumita did this, and then you can see here, in collaboration with Australia, of course, because we do not have a very good data analyzer here. Sequencing can be done. This was a whole genome sequencing approach, or the whole genome sequencing approach, and then that can be done. But to read the data, we do not have a data analyzer. So, with the help of Australian data analyzer, we read that and we found that these are two flanking genes, and there was no modifications in the expression of the flanking gene, the same expression. Control and weakness. Okay, so that is a big answer to the regulators, and we are hopeful that regulators will be happy to, because this you have to show before the field test. Once you want to take to the field test, you have to show all these things. Okay, you also have to show that there is no alteration in the nutritional quality of the seed. Chickpea has a specific nutritional quality, right? But that cannot be, that should not get modified, So one of our PhD students, Ruby Gupta, she showed that that she did a lot of experiments. I'm not going to discuss those things in detail here, but she confirmed that no significant difference in primary nutritional compositions, fatty acids, isoflavones, etc., was recorded between transgenic and non-transgenic chickpea. Is a message. Okay? And she published this in 2020. Okay. Now you see, we transfer to different national institutions. You see, being at northeastern region, you don't feel there are many of us outside northeast, but mostly we are from northeast here, right, in this particular seminar. Okay? Rahul bhai, तुम लोग राहुल भाई ट्रेनिंग दे रहे ना, वो सोचते हैं कि ये बड़ा रिमोट प्लेस है. Before coming here, he thought Assam is quite a jungles and all. और सांप निकलते हैं यहाँ पे, tigers निकलते हैं, ऐसे सोचते हैं. But they always have feelings. Till now, till today they have those feelings. But we have to show that we are not at remote place. By our work, by our efficiency, we have proved that. Okay. So you see, today this technology has gone to many national institutions, but at the same time, now Bangladesh has showed their interest. Bangladesh government has showed their interest, and we have developed a project with Australian government, Bangladesh, and then Assam Agricultural University. We visited Bangladesh for seeing the chickpea cultivations there, for interacting with the farmers there, and then you see farmers were so excitedly they were looking at us, that these are the gods. They were bringing the chickpea insect resistant chickpea to us. Okay. So now the program is going on, and I'm not sure whether Indian farmers will get the chickpeas, Tanzanian chickpeas before Bangladesh, or Bangladesh will get before Indian farmers? It's a big question. Because with the brinjal, you know, right? With the brinjal case, same Mahiko, Maharashtra Seed Company developed with the brinjal, resistant to fruit and sugar borer. And Indian government did not allow, because there was big hue and cry, public opinion was taken. And you all know that there was a monetary. But at that time, Maharashtra Seed Company, the same BD, brinjal, they took to Bangladesh upon invitation in 2010. And in 2013, that BD brinjal was, released in Bangladesh. At that time, initially 20 farmers cultivated BD brinjal, and today more than 50,000 farmers are cultivating. Talking about more than 10,000 hectares of it. So that's the situation there. So anyway, means technology may be transferred to international organization as well. Okay, now what happens here? Apart from transgenic, Sumita is going to actually take a class, right? Sumita, are you going to take a class on everything of chickpea? So this is just I'm mentioning here. As I say, I mentioned about Sumita's experiment and the program project. Okay, what she is trying to make, the genome editing, the optimized genome editing method for chickpea, which CRISPR-Cas9 system to edit that, AHAS1 gene, that is, what it was, alcohol, acetyl hydrogen, acetyl hydroxyl acetic, acetyl hydroxy acetic acid synthase, right? No, acetyl hydroxy acetyl synthase. Okay. Acetyl hydroxy acetyl synthase. So that gene, you see, what happens is when you apply herbicide, this particular gene, encodes that protein, and herbicide actually, binds to that, protein. And this protein this enzyme is responsible for, encoding the essential amino acids. Okay. So, unless these amino acids are produced, the plants cannot grow. Right? So, what happens when we develop the plant with this particular, editing system? What she is going to do now, she is going to edit this gene. Just change of one or two base pairs. Then this particular enzyme will not get produced. So herbicide cannot take up. That is the story. She is thinking, she is writing the story now, and she is working with the story in the lab. And I'm hopeful that in next couple of years or at least 3-4 years, she will be able to develop. Because you can see here, she has already developed the system, transformation system with the genome edited, construct, and then she has already developed some of the lines, and these lines are showing PCR positive. So she has to go much more, from these. But at least in the initial experiments, she has confirmed that gene has, getting, going into the system. Okay. She has confirmed that whether the specific gene got knocked down or not. That she has to. Okay, just 5 minutes more. I have to cross the time. So do you have another concept here? Sorry, I will need another 5 minutes. Huh? It's a hands-on, no? So I can take, right? 2 minutes, you function. Can I go for another 10 minutes? Okay. So you see, should we say yes to the genetically engineered crops now? That's the big question. How many of you would like to oppose now? Students for a while. Okay. Just a bit of water. Is there anyone to oppose? Is there anyone to oppose? Obviously, my students or our students, say, well, they will be famous. Brilliant. This guy, what he can do, you don't know. So, outside, somebody is saying, you have nothing to get scared. Do you really want to support the GM, or you have some oppositions? Okay?
student, the person who is speaking, he ate BT brinjal. So he
So anyway, so you see, let us see, whether they are globally accepted. That's what we have to see. Okay, now you see, why it is not moving? Some big opposers are there, I think. That's why this is, you see, magic. It's not moving. Really, it's not moving. What happened? It's not moving. Right? Apa kau boleh beli? Tak ada. Some boleh nuang. Shortcut is yes. Mana kau ni?
Oh, hammer is what level?
Ada tu apa hammer is? Ini bisa ni kan musuh. Bukan nama apa dah sih. Mana boleh bisa? Oh, ini kan remote apa buku apa ni? Itu mau remote apa ni? Remote apa boleh ni? Jadi laptop dah hang mah. अच्छा खाना तो क्या बन गया सर सुनिए वो बाहर में खाली खाना बड़ा बोल रहे हैं मांस को सब अच्छा है दिसे ठीक है मांस दिसे अरे दिसे मांस चिकन 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 बड़ा है और राहुल भाई तुमने क्या है हाँ पनीर पनीर के मिला चिकन के मिला मछली के मिला Well, and then, I think I have seen you before. You are from this department, right?
Yes, sir. Yes, sir. I was project.
Oh. What was your name? Modumita. Modumita. Yeah, you see, I remember. I should be awarded. Normally, my mother sometimes she say, you may forget sometimes your own name. Because I'm so poor in remembering names. Okay. But I don't know how did I remember. Modumita. Actually, my wife name is Modumita. Similarly, what happens?
Jadi ulang lagi gitu. Tuh ya gitu. Memang perlu lagi. Thank you.